Hello, dear students. Welcome to our topic about transcription and translation. In this topic, you will learn how the DNA inherited by an organism leads to specific traits. Before I talk about transcription and translation in detail, let me first give you an overview. First, you need to understand the flow of genetic information. The information content of DNA is in the form of specific sequences of nucleotides, as what I've mentioned in my previous discussions. Now, the DNA inherited by an organism leads to specific traits by dictating the synthesis of proteins. Proteins, therefore, are the links between genotype and phenotype. As mentioned, proteins are the link between genotype and phenotype, or basically the expression of traits. This process is known as gene expression. This is a process by which DNA directs the synthesis of proteins, and the expression of genes that code for proteins includes two stages: transcription and translation. Genes provide the instructions for making specific proteins, but a gene does not build a protein directly. The bridge between DNA and protein synthesis is the nucleic acid RNA. This process was dubbed as the central dogma by Francis Crick in 1956. Transcription is the synthesis of RNA under the direction of DNA. Transcription produces messenger RNA or mRNA. Meanwhile, translation is the synthesis of a polypeptide using the information in the mRNA. Translation happens within the ribosomes. In prokaryotes that are devoid of the nucleus, translation of mRNA can begin before transcription has finished. However, in a eukaryotic cell, the nuclear envelope separates transcription from translation. After transcription, eukaryotic RNA transcripts are modified through RNA processing to yield finished mRNA. A primary transcript is the initial RNA transcript from any gene prior to processing. We have here a figure to differentiate the transcription and translation processes between prokaryotes and eukaryotes. In a prokaryotic cell, in this example a bacterial cell which lacks a nucleus, mRNA produced by transcription is immediately translated without additional processing. However, in a eukaryotic cell, the nucleus provides a separate compartment for transcription. The original RNA transcript, called pre-mRNA, is processed in various ways before leaving the nucleus as mRNA. Now you might be wondering how the instructions for assembling amino acids into proteins are encoded into DNA when there are 20 amino acids, but there are only 4 nucleotide bases in DNA. This leads us now to the genetic code. Triplets of nucleotide bases are the smallest units of uniform length that can code for all the amino acids. These triplets of nucleotides are called codons. The flow of information from gene to protein is based on a triplet code, which is a series of non-overlapping 3 nucleotide words. The words of a gene are transcribed into complementary non-overlapping 3 nucleotide words of mRNA. These words are then translated into a chain of amino acids, forming a polypeptide. We have here an illustration of the triplet code. During transcription, one of the 2 DNA strands, called the template strand, provides a template for ordering the sequence of complementary nucleotides in an RNA transcript. The template strand, by the way, is always the same strand for a given gene. Now, during translation, the mRNA-based triplets, called codons, are read in the 5' to 3' direction. Each codon specifies the amino acid to be placed at the corresponding position along a polypeptide. Molecular biologists cracked the genetic code of life in the early 1960s when a series of elegant experiments disclosed the amino acid translations of each of the RNA codons. By mid-1960s, all the 64 codons were deciphered. 61 code for amino acids, while 3 are stop signals to end translation. The genetic code is redundant, which means that more than one codon may specify a particular amino acid, but not ambiguous, meaning to say that no codon specifies more than one amino acid. Codons must be read in the correct reading frame or correct groupings in order for the specified polypeptide to be produced. This figure shows the codon table for mRNA. The 3 nucleotide bases of an mRNA codon are designated here as the first, second, and third bases, reading in the 5' to 3' direction along the mRNA. Now let's take the codon AUG as an example. The codon AUG not only stands for the amino acid methionine or MET, but also functions as the start signal for ribosome to begin translating the mRNA at that point. 3 of the 64 codons function as stop signals, marking where ribosomes end translation. Now let me talk about the process of transcription or the synthesis of RNA under the direction of DNA. Let us first talk about the molecular components of transcription. First on the list is RNA polymerase. This enzyme catalyzes RNA synthesis and pries DNA strands apart and hooks together RNA nucleotides. Please take note that the RNA transcribed is complementary to the DNA template strand. RNA synthesis follows the same base pairing rules as DNA, except that uracil substitutes for thymine, meaning to say, cytosine binds with guanine and adenine binds with uracil. Specific sequences of nucleotides along the DNA mark where transcription of a gene begins and ends. The DNA sequence where RNA polymerase attaches and initiates transcription is known as the promoter. In bacteria, the sequence that signals the end of transcription is called the terminator. And the stretch of DNA that is transcribed into an RNA molecule is called a transcription unit. The synthesis of an RNA transcript involves 3 stages. First is initiation, followed by elongation, and lastly, termination. So now we go to RNase polymerase binding and initiation of transcription. So promoters initially signal the transcriptional start point and usually extend several dozen nucleotide pairs upstream of the start point. Special proteins, known as transcription factors, mediate the binding of RNA polymerase and the initiation of transcription. The completed assembly of transcription factors and RNA polymerase bound to a promoter is called a transcription initiation complex. A promoter called a Tata box is crucial in forming the initiation complex in eukaryotes. This figure explains the terms previously mentioned. A eukaryotic promoter commonly includes a Tata box, which is a nucleotide sequence containing TATA. This is the site where RNA polymerase attaches. However, several transcription factors need first to recognize the Tata box and bind to the DNA before RNA polymerase can bind in the correct position and orientation. Then more transcription factors bind to the DNA along with RNA polymerase, forming the transcription initiation complex. RNA polymerase then unwinds the DNA double helix, and RNA synthesis begins at the start point on the template strand. Next to initiation is the elongation of the RNA strand. Now, vital in elongation is still the RNA polymerase, which untwists the double helix 10 to 20 bases at a time. A gene can be transcribed simultaneously by several RNA polymerases, and the nucleotides are added to the 3' end of the growing RNA molecule. Transcription progresses at the rate of 40 nucleotides per second in eukaryotes. During elongation, RNA polymerase moves along the DNA template strand, joining complementary RNA nucleotides to the 3' end of the growing RNA transcript. Behind the polymerase, the new RNA peels away from the template strand, which reforms a double helix with a non-template strand. And then the last stage of transcription is termination. The mechanisms of termination are different in bacteria and eukaryotes. In bacteria, the polymerase stops transcription at the end of terminator, and the mRNA can be translated without further modification. However, in eukaryotes, RNA polymerase transcribes the polyadenylation signal sequence that codes for a polyadenylation signal, which is composed of the nucleotides AAU, AAA, and then the RNA transcript is cut free by proteins past this polyadenylation signal. After the termination of transcription, a pre-mRNA transcript or a primary transcript is formed. The pre-mRNA transcript, before it is released into the cytoplasm as mRNA, undergoes RNA processing. During this time, enzymes in the eukaryotic nucleus modify pre-mRNA before the genetic messages are dispatched to the cytoplasm. In RNA processing, both ends of the primary transcripts are altered and some interior parts of the molecules are cut out and the other parts are spliced together. One of the ways in how the primary transcript or the pre-mRNA molecule is modified is through alteration. Each end of a pre-mRNA molecule is modified in a particular way. The 5' end is synthesized first. It receives a 5' cap, which is a modified form of a guanine nucleotide added onto the 5' end after the transcription of the first 20 to 40 nucleotides. Meanwhile, the 3' end of the pre-mRNA molecule is also modified before the mRNA exits the nucleus. An enzyme adds multiple adenine nucleotides to the 3' end, forming a poly-A tail. The poly-A tail is composed of 50 to 250 adenine nucleotides added to the 3' end. These pre-mRNA modifications share several important functions. First, they facilitate the export of the messenger RNA, and then they protect the messenger RNA from hydrolytic enzymes. Lastly, these modifications help ribosomes attach to the 5' end. Now let us talk about split genes and RNA splicing. Most eukaryotic genes and their RNA transcripts have long non-coding stretches of nucleotides that lie between coding regions. These non-coding regions are called intervening sequences or introns. The other regions are called exons because they are eventually expressed and translated into amino acid sequences. Now, the process of RNA splicing removes introns and joins exons, creating an mRNA molecule with a continuous coding sequence. Let us take this RNA molecule as an example. The RNA molecule shown here codes for beta-globin, which is one of the polypeptides of hemoglobin. The numbers under the RNA refer to codons. Beta-globin is 146 amino acids long. The beta-globin gene and its pre-mRNA transcript have 3 exons, which correspond to sequences that will leave the nucleus as messenger RNA. During RNA processing, the introns are cut out and the exons are spliced or joined together. This is the process of RNA splicing. But how is RNA splicing carried out? Small nuclear ribonucleoproteins, abbreviated as SNRNPs and pronounced as SNRPs, are located in the nucleus of the cell and are composed of RNA molecules. SNRPs recognize splice sites and join with other proteins to form a spliceosome. The spliceosome interacts with certain sites along an intron, releasing the intron, which is rapidly degraded, and joining together the 2 exons that flanked the intron. Another important molecule that contributes in RNA splicing are ribozymes. These are catalytic RNA molecules that function as enzymes and can splice RNA. So now let us proceed to the second stage of gene expression, which is translation. Genetic information flows from messenger RNA to protein through this process of translation. So before I explain to you the important steps of translation, let me first give you the molecular components of translation. So first is the transfer RNA or tRNA. tRNA helps translate an mRNA message into protein, and tRNA also transfers amino acids to the growing polypeptide in a ribosome. Translation is a complex process in terms of its biochemistry and mechanics. This figure shows the overview of the translation process. As a molecule of mRNA is moved through a ribosome, codons are translated into amino acids one by one. The interpreters are tRNA molecules, each type with a specific anticodon at one end and a corresponding amino acid at the other end. A tRNA adds its amino acid cargo to a growing polypeptide chain when the anticodon hydrogen bonds to a complementary codon on the mRNA. Since tRNAs are important in the translation process, it is just appropriate that we review its structure and function. So molecules of tRNA are not identical. Each carries a specific amino acid on one end, and each has an anticodon on the other end. The anticodon base pairs with a complementary codon on mRNA. As mentioned in the short discussion about the RNA, a tRNA molecule consists of a single RNA strand that is only about 18 nucleotides long. The two-dimensional structure of a tRNA resembles a clover leaf. It is flattened into one plane to reveal its base pairing. And because of hydrogen bonds, tRNA actually twists and folds into a three-dimensional molecule. Now, an accurate translation process requires two steps. First, there must be a correct match between a tRNA and an amino acid. This is done by the enzyme amino-acyl tRNA synthetase. Second, there must be a correct match between the tRNA anticodon and an mRNA codon. This figure shows the enzyme amino-acyl tRNA synthetase joining a specific amino acid to a tRNA. So first, active site binds the amino acid and ATP. So the linkage of a tRNA and amino acid is endergonic process that occurs at the expense of ATP. The ATP loses two phosphate groups, becoming adenosine monophosphate. Then, appropriate tRNA covalently bonds to amino acid, displacing adenosine monophosphate. And then lastly, the tRNA charged with amino acid is released by the enzyme. Another molecular component that is also important for translation are ribosomes. Ribosomes facilitate specific coupling of tRNA anticodons with mRNA codons in protein synthesis. So the ribosomal subunits, both large and small, are made of proteins and ribosomal RNA. The ribosome has three binding sites for tRNA. First is the P site or the peptidyl tRNA binding site. This holds the RNA that carries the growing polypeptide chain. Next is the A site or the amino-acyl tRNA binding site. The A site holds the tRNA that carries the next amino acid to be added to the next chain. And lastly, the E site or the exit site. This is where discharged tRNAs exit or leave the ribosomes. Similar to transcription, translation also has the following stages. First, initiation, followed by elongation, and lastly, termination. All the three stages require protein factors that aid in the translation process. For certain aspects of chain initiation and elongation, energy is also required. It is provided by the hydrolysis of guanosine triphosphate or GTP, which is a molecule closely related to ATP. So now we move on to the first stage of translation, which is initiation. The initiation stage of translation brings together messenger RNA, a transfer RNA with the first amino acid, and the two ribosomal subunits. First, a small ribosomal subunit binds with a messenger RNA and a special initiator transfer RNA. Then the small subunit moves along the messenger RNA until it reaches the start codon, which is AUG or methionine. A small ribosomal subunit binds to a molecule of messenger RNA. In a bacterial cell, the messenger RNA binding site on the subunit recognizes a specific nucleotide sequence on the mRNA just upstream of the start codon. An initiator tRNA with the anticodon UAC base pairs with a start codon AUG. This tRNA carries the amino acid methionine. The union of mRNA, initiator tRNA, and a small ribosomal subunit is followed by the attachment of a large ribosomal subunit, completing the translation initiation complex. Proteins called initiation factors bring in the large subunit that completes the translation initiation. Next to initiation is the elongation stage. During the elongation stage, amino acids are added one by one to the preceding amino acid at the C terminus of the growing chain. Each addition involves proteins called elongation factors and occurs in three steps. First, codon recognition, then peptide bond formation, and lastly, translocation. Translation proceeds along the mRNA in a 5' to 3' direction. This figure shows the three steps involved in the elongation process of translation. First is codon recognition. The anticodon of an incoming amino acid tRNA base pairs with a complementary mRNA codon in the A site. Hydrolysis of GTP increases the accuracy and efficiency of this step. Next is peptide bond formation. An rRNA molecule of the large ribosomal subunit catalyzes the formation of the peptide bond between the amino acid group of the new amino acid in the A site and the carboxyl end of the growing polypeptide in the P site. This step removes the polypeptide from the tRNA in the P site and attaches it to the amino acid on the tRNA in the A site. And lastly is translocation. The ribosome translocates the tRNA in the A site to the P site. At the same time, the empty tRNA in the P site is moved to the E site where it is released. The mRNA moves along with its bound tRNAs, bringing the next codon to be translated into the A site. Next to elongation is termination. So the termination of translation occurs when a stop codon in the mRNA reaches the A site of the ribosome. So the stop codons are the following: UAG, UAA, and UGA. The stop codons do not code for amino acids, but instead act as signals to stop translation. The release factor causes the addition of a water molecule instead of an amino acid. This reaction releases the polypeptide, and the translation assembly then comes apart. This photo shows the termination of translation. When a ribosome reaches a stop codon on mRNA, the A site of the ribosome accepts the release factor. The second figure shows the release factor promoting the hydrolysis of the bond between the tRNA in the P site and the last amino acid of the polypeptide, thus freeing the polypeptide from the ribosome. Lastly, the two ribosomal subunits and the other components of the assembly dissociate. Now, let us talk about polyribosomes. So, a number of ribosomes can translate a single mRNA simultaneously, forming a polyribosome or polysome. So, polyribosomes or strings of ribosomes enable a cell to make many copies of a polypeptide quickly. Often, translation is not sufficient to make a functional protein. That is why polypeptide chains are modified after translation or targeted to specific sites in the cell. So, which leads us now to this subtopic on completing and targeting the functional protein. One of the ways in which proteins are modified after translation is by protein folding. So, during and after synthesis, a polypeptide chain spontaneously coils and folds into its three-dimensional shape. Proteins may also require post-translational modifications before doing their job. Some polypeptides are activated by enzymes that cleave them, and also other polypeptides come together to form the subunits of a protein. Next is targeting polypeptides to specific locations. So there are two populations of ribosomes that are evident in cells: free ribosomes and bound ribosomes. So as a review, free ribosomes mostly synthesize proteins that function in the cytosol, and then bound ribosomes make proteins of the endomembrane system and proteins that are secreted from the cell. So ribosomes are identical and can switch from free to bound. Polypeptide synthesis always begins in the cytosol, and the synthesis finishes in the cytosol unless the polypeptide signals the ribosome to attach to the endoplasmic reticulum. But what really determines whether a ribosome is free in the cytosol or bound to the rough endoplasmic reticulum? Now, polypeptides destined for the endoplasmic reticulum or for secretion are marked by a signal peptide. A signal recognition particle or SRP binds to the signal peptide. The SRP brings the signal peptide and its ribosome to the endoplasmic reticulum, meaning to say the SRP or the signal recognition particle functions as an escort that brings the ribosome to a receptor protein built into the endoplasmic reticulum membrane. This picture shows the signal mechanism for targeting proteins to the endoplasmic reticulum. A polypeptide destined for the endomembrane system or for secretion from the cell begins with a signal peptide, which is a series of amino acids that targets it for the endoplasmic reticulum. This figure shows the synthesis of a secretory protein and its simultaneous import to the endoplasmic reticulum. In the endoplasmic reticulum and then in the Golgi, the protein will be processed further. Finally, a transport vesicle will convey it to the plasma membrane for release from the cell. So this ends my discussion on transcription and translation. I hope you guys have learned something. Thank you for watching this video.